Today's topic is flow cytometry. So what is flow cytometry? Flow cytometry is made up of three words. Flow, that is something is flowing, the fluid is there because it is using the hydrodynamic mechanism for focusing the particular sample of the containing the cells. Cyto means something related to cells, and metry means measurement. We are counting the number of the cells. So it is a laser-based test. Flow cytometry is a laser-based test which will detect the chemical and physical differences of the cells or particles. And we can use bone marrow or peripheral blood sample for its analysis. So what is the process in flow cytometry? Flow cytometry is a fluorescent technique used to identify and enumerate the cells pairing particular antigen or antigens or surface markers by suspending them in a stream of fluid passing through the electronic detection apparatus. And during the process, the cells or particles are first suspended in a fluid and they are tagged with a fluorescent activated cell sorting labels. So this is called as fluorescent activated cell sorting. The fluorescent antibody dyes are used which will label or which will tag those particular things inside or on the surface of the cells. The cells are then injected in the flow cytometer machine, and the computer can analyze the multiparametric analysis, and the process approximately 10,000 cells under a minute can be calculated. So what are the parameters you can see here? Size, granularity, RNA DNA content, cellular antigens, receptor levels. All these are the parameters which are taken into the consideration for identification of the cells, counting the cells. Now, the cells are made to flow in the single stream by hydrodynamic focusing. So hydrodynamic focusing I will show you in the schematic diagram. And there is a surrounding layer of the sheath fluid. Sheath fluid is also there. And argon laser about 488 nanometer frequency is focused on the cells, and various sensors they pick up the laser light reflected by the cells, and the parameters are measured by flow cytometer. There are two types of scatters. One is a forward scatter FS and side scatter. Side scatter will detect the granularity of the cells. The emitted light is directed into the various sensors by combination of the filters. Now this is the schematic diagram. So this is outermost fluid, that is called a sheath fluid, and this is the sample port where the samples are added, but before adding the sample it is treated with fluorescent substance. So the stained cells in the suspension are there, are used, and you can see there is a hydrodynamic focusing of the cells which will pass through the single file, and then there is a laser light source, and the fluorescent emitted from the cells, they will scatter it. So SS and FS. For FS is for forward scatter and SS is for side scatter. So it will analyze the pattern of the scatter, and depending upon that what are the cells, the counting of the cells is done. Now you can see this is the scatter pattern. The blue color one is shown as a monocyte pattern, and you can see the lymphocyte green, and in the center there is a yellow dot. So you can see this scatter pattern is there, but we have nothing to do with it because electronically this is analyzed by the equipment. It will give the counts of the cells. So this is the machine, you can see. So what are the parameters measured? The parameters measured are size, then granularity, DNA RNA content, cellular antigens, and receptor levels. So these are the various parameters used to analyze the number of the cells. So what are the applications? So counting and cell sorting. So all of you know DLC, differential leukocyte count is there. Then determining the cell characteristics and functions, that is also another use. Then detection of diagnosis and treatment of blood and bone marrow. Prognosis also can be done by again repeated testing, finding the biomarkers, a measurable substance whose percentage might predict the cancer behavior or response to the treatment. Then immunophenotyping can be done, and CD4 and CD8, it's well known. CD4 count is well known for prognosis of HIV positive individuals. Thank you for your patient listening. Thank you.